Sir belongs from Tamil Nadu and specialized in tree breeding center and ICR. And sir has been recognized Indian Society of Agroforestry. Sir has got Fellow Award by Indian Society of Agroforestry and is Eminent Scientist Award by Society of Science of Climate Change and Sustainable Environment. Sir has more than 30 research papers and has guided 4 MSc students and sir has attended more than Sir, please start your presentation. The title for the presentation is going to be on genetic resource and marker assisted breeding for enhancing productivity of MPTS in agroforestry.
Thank you very much for your introduction and welcoming me. So, can you please help me to share my slides? I'm unable to load my slides.
Yes, sir, you can click on share screen. Then click on share entire screen. Sir, are you able to do it?
Yeah, just it got enabled. Thank you. I hope my presentation is visible, right?
No, sir, it's not visible. You just have to click on share entire screen.
Ma'am, it is visible. It's visible. It's visible. I think your net connectivity is low.
Yeah, maybe. Sir, please go ahead then if it is visible to the audience.
Okay, thank you very much. Yeah, so I'm here to present about tree genetic resources and marker assisted selection for various multi-purpose tree species. So I don't want to go much on agroforestry definition and all, as you know all. So although I would like to give some touch on that, agroforestry deals about trees, crops, and livestock together for sustainable land use system and production. And as of now, more than 100 distinct forms of agroforestry are practiced worldwide, with farmers integrating tree shrubs among annual herbaceous crops. And as so far recognized worldwide as agroforestry is an integrated approach for sustainable land use system. And coming to the importance of agroforestry, we know that the agroforestry is offering so much of benefits like improved nutrition and food security and more benefits from trees, and it creates resilience to various livelihoods, security, and climate change mitigation and adaptation. It offers many environmental benefits, support local communities, and restoring various cultural practices which have been followed. And ultimately, the aim of agroforestry is to reduce the poverty. And yeah, and coming to the advantages of trees, as we know, the trees offers multiple benefits to humans, animals, crops, and many living organisms on the earth. And it can also used as a source for fuel, food, and non-wood products as well. It provides nutritious food, nuts, leaves for consumption in household purpose. And these trees are the residues can be used as wood energy for cooking and heating, while leaves can be used as forage for many livestock. Of course, we would like to know before entering in the subject, the trees possess, because trees offers multiple benefits. So these benefits are obviously coming from because of its characteristics features. So these trees are having ultimate characters like its perennial growth habit, woody morphology, slow growth, and long juvenile period, Mostly these trees are outcrossing behavior and holds high heterozygosity genetic makeup, relatively high genetic diversity as well. And for each tree, holds seed to seed generation cycle is varies and it's large usually. And ultimately, the high outcrossing behavior and the heterozygosity behavior leads to high inbreeding depression as well in the trees. And tree genetic resources. What is tree genetic resources? Tree genetic resources are the heritable material which holds the sum of superior alleles among the species or within the species, and these genetic materials provides goods and services which are essential for human well-being and sustainable development from time to time. And most tree species have high levels of genetic diversity, offering great opportunities for growing, selecting, and breeding trees for various end users. And priorities of tree genetic resources. As we know that the tree genetic resources are very important in improving the availability of and to access the information on tree genetic resources for various end users. And these tree genetic resources are being conserved in situ as well as ex situ. And also, as I earlier said, these tree genetic resources are much useful in sustainable use, development, and management of tree genetic resources for various economic trait improvements according to human needs. And of course, the major priority of TGR lies in various policies and institutional level and capacity buildings. And what are all the focused objectives on tree genetic resources? So the objectives are mainly the tree genetic TGR objectives are exploration, collection, and conservation of various MPT gem blossoms, and conservation of these tree genetic resources. Either it can be conserved in the field gene bank, or it can be conserved as DNA or any organelle in the organelle or DNA bank. And then the conserved genotypes can be further assessed for its genetic diversity and variation for its economic traits, and also characterization and identification of high oil yield, fast growth, wood properties, etc., depending upon the trait of interest from the species. And also, the tree genetic resources are ultimately useful in genetic improvement and production of improved planting material for sustainable production and productivity. And coming to the tree breeding and its prospects, so tree breeding involves the application of genetic principles for the mass production of seedlings with desired traits in order to achieve higher productivity, better adaptability to various environment in the changing climatic scenario as well. And the main prospects of tree breeding is to reduce the cycle, to reduce the harvesting cycle. This is one of the main prospects of why tree breeding is carrying, because as we know that the trees are having long germinal period, and also the harvesting cycle also varies from tree to tree, and it's almost it's highly it's perennial in nature, so the availing the end use will take too much of time. So ultimately, the tree breeding will enhance its yield per hectare and reducing the unit cost of the wood or whatever the economic trade may boost up sustained commercial forestry in the near future. So these are some of the prospects why tree breeding attracts. So, as we know that the tree breeding earlier days, the tree breeding the only means is conventional breeding. Although the conventional breeding is very important and it has its own demerits, so that particularly the demerits like it's the lengthy time process and resources uh, and the accuracy, the trait accuracy, because the conventional breeding many times influenced by the environmental factor, and that thereby the accuracy of the conventional breeding is compared to the marker assisted breeding is still lesser. So with the advent of marker technology, these constraints has been eliminated and becomes kind of fast track mode of tree breeding for various trait improvements. So coming to the markers, so generally three types of markers are there: morphological marker, biochemical marker, and molecular marker. Morphological markers, as we know that whatever the visual what we are seeing in the field on the tree, individual trees, it's like a appearance, like a morphological characters like its color, shape, size, everything comes under morphological variation. And these variations are, it's a kind of it gives a large effect variation because these mostly relies on the qualitative trait in nature. And coming to the biochemical markers, isocyans are the best example of biochemical markers. Before DNA markers, these biochemical markers were played a major role in tree as well as crop improvement. And later, the molecular markers are evolved with great scope for genotyping various trees. And these various types of molecular markers are there. These molecular markers are usually we used to call as DNA markers, broadly divided into two. One is a restriction digestion based, another one is PCR based marker. Say, for example, restriction digestion RFLP are the example for the restriction digestion based marker system, and RAPD, SSR, ISSR, SNP. These are PCR based markers involved in marker assisted selection. And these are different DNA markers used in tree breeding so far with its characters. So if you see that particularly the kind of RABD markers are used to give genomic abundance because of its high genomic abundance. It gives more information, particularly on its diversity evolution studies. RAPD markers are highly useful. And whereas coming to SSR and SNP markers are also its wide occurrence among the genome, and thereby it gives more and more accurate information on its genotypes of different trees. And these are some of the primary application depending upon the marker we have quoted. Say, for example, RFLP is involved in genetic information, linkage map. RAPD is highly involved in diversity assessment and evolution and phylogenetic relationship studies. And if you see SSR and SNPs, you can almost all the purposes. It includes diversity, genetic information study, as well as evolution purpose. So these are some of the informations on different markers used in tree breeding. And coming to the applications of molecular markers, these molecular markers are major application in estimating or assessing the genetic diversity, and it's highly helpful in genome mapping, gene pyramiding, and assessing and identifying the hybrid purity, and also identification of superior parent line for future hybridization programs. And more importantly, it is also involved in DNA fingerprinting for varietal identification, cultivar identification as well. These are the major applications of marker assisted selection in tree breeding. And this is a schematic representation of how marker assisted breeding is going to carried out in multi-purpose tree species. Here you can see the parent one and parent two. Here parent one is a susceptible or a kind of recurrent parent. Parent two is a donor parent. And you see the in the from the donor parent, this is our desirable allele. And this allele, ultimate our aim is to bring this allele in the background of its recurrent parent. So both the parents are crossed and F1 we are developing. And in these efforts are further selfed to make F2 population or advanced breeding population. From this population, we can take DNA from any part of the tissue from these plants, and we can go for genotyping with the polymorphic markers which we discussed earlier. And with these markers, we can able to identify the target, our gene of interest or our target trait has been present in the progenies in the very early stage. You can see here, this figures shows that carries its target allele, and which are free from the undesirable allele or any kind of linked traits. So by the way, the selection is more accurate, and these selection are more accurate with it's in a short time, so that this will accelerate the breeding process to develop any variety or any cultivar. And this is one of the case study in Apple. If you see that in this A, this figure, it's a mode of conventional breeding. This B part, it shows fast track breeding or marker resisted breeding. You see how the conventional breeding takes time to transfer a trait. It takes several, it involves several backcross, 4 to 5 backcrosses involved, and to select a mapping population itself 4 to 5 years. Ultimately, it's in 20 to 24 years it will take to test or to carry forward the trials further for commercial use. Whereas in the marker assisted breeding, you can see here the disease-resistant wild species is crossed with a transgenic line or a recurrent line which carries a gene called BPMADS4. These genes are known. The role of this gene overexpression is to reduce the flowering period. So, by crossing this in the second year itself, we can assess by marker assisted breeding whether the target gene is present or not. So once the target gene is we have identified, then this we can use it as a kind of a donor parent and can be crossed with a superior quality cultivar to transfer the target trait to the superior cultivar in within a year or 2 years we can transfer, and then with the help of molecular markers we can identify precisely, we can identify our gene of interest, and next 4 to 5 years we can develop lines that can be further forwarded for testing and approval for commercial use. So you can see the time difference that in a conventional mode, yeah, usually 20 to 25 years where it's taking. In marker assay selection, we can go with only 4 to 5 years. We can able to transfer our target gene with more precisely. So this is the major advantage of marker assisted selection, and more importantly here, the linkage drag. It means that undesirable effect of the genes which are linked with our target gene. These limitations are also can be eliminated in the marker assisted breeding. So coming to the applications of genomic technologies in various multi-purpose tree species, the major application of genomic technology is bridging the gap between phenotype to genotype. This is very much important because when we bridging the gap, we will come to know that we will tightly link the phenotype with the genotype. So thereby one can accurately map, or one can accurately genotype on the trait of interest, and it can be later with the help of functional markers can be transferred. So in that way, similarly, the genetic resources like germ plasm plus tree provenances or any elite varieties can be genotyped or sequenced with the help of high throughput platforms. So and then based on the reference genome, this can be sequenced, and then if parallelly these, if suppose where the reference genomes are well, it is available, although the genome information available, once again we can resequencing of these genomes in order to make a fine tune of the genes and its precise positions of various desirable alleles. And also apart from that, various genotyping platforms such as like marker based or array based platforms are available, and these platforms can be effectively put into the genetic resources for identify and transfer the trait of interest. And more importantly, functional omics such as transcriptomes, metabolomics, proteomics are being used to characterize the candidate genes, and superior halophytes can be identified, and these can be effectively linked with the phenotype with the appropriate kind of statistical analysis, and this will help in assessment of genetic diversity, where the marker information, where the prior information, also where it is not available. And of course, these all the genomic resources, the information, whatever we are gaining, that can be put it back in the marker assisted backcrossing, and according to our trait of interest for fast track breeding. So all these together will enhance or accelerating our tree breeding process to develop an elite cultivar. Either it may be for climate adaptive trait, or it may be for quality improved, any quality improved like a wood quality, or it may be yield improvement for oil or fruit yield, whatever it may be. So these things can be done with the application of genomic technologies aided with molecular markers. And coming to the genotyping platforms, as we know that the advent of various technologies, the emerging sequencing technologies, various sequencing technologies also evolved. So these one of the these sequencing technologies may offer useful markers for genotyping effectively, and one of the SNPs are found to be the most effective marker from various sequencing technologies. And these markers, because wide occurrence and evidence availability and coverage information is more across the genome. So one can choose this SNP for genotyping depending upon their objectives as well. So for example, high density genotyping platforms are mostly used for genome-wide association studies as well as linkage mapping. And then medium density genotyping platforms can be used for genetic diversity as well as genomic selection studies. And similarly, low density genotyping studies can be used for marker assisted backcrossing and gene tagging studies. So, depending upon the objectives, one can choose the genotyping platform. These are the developments with the advent of next generation sequencing technologies. And coming to the sequence-based holistic tree breeding approach, as we earlier discussed, any kind of mapping population or genetic resources can be deployed with functional omics tools, and that functional omics tools may offers genome-wide association studies, and it can help in identification of superior genes and alleles in the population. And apart from that, the high throughput sequencing combined with multi-location phenotyping can also aid in a genome-wide association study to identify the superior alleles. And also, when all together these superior genes and alleles, GWAS, can be directly deployed into a breeding population for effective identification of parental selection, and thereby early generation screenings are possible to move further to high genomic estimated breeding values. And not only here, yeah, one can directly from high throughput sequencing and multi-location phenotypic data can directly go for using predictive models. They can identify high genomic estimated breeding value lines, and these lines can be very rapidly can make further forward for multi-location yield trials, and ultimately it can be released as an improved variety for our target trait. And these are some of the examples of complex traits. Although there are many complex traits identified in recently identified in trees, I have quoted some of the QTLs in various MPTs for the traits like growth, wood properties, biotic stress, abiotic stress, and all these QTLs. The purpose of identifying and dissecting the QTLs is ultimately to introgress into the elite cultivar for improving economic yield or economic end-use traits. Also, it involves in quality improvement and resistance to stresses like biotic and abiotic, because we know that most of the traits, economic traits, are governed by complex nature and controlled by many genes. So these using gene pyramiding approach or marker assisted backcrossing, one can precisely, rapidly can QTLs can be introgressed into and target cultivars to produce a light or an improved cultivar. And coming to the sequencing approaches, also the sequencing approaches can be used to decode the coding and non-coding elements. Here in this picture, if you see that these germplasm zones can be with the use of advent of NGS, one can go for either structural genomics or one can go for functional genomics by transcriptome. So through genome, one we can able to identify the non-coding DNAs like randomly repeated DNA, transposable elements, and spacer DNA. And similarly, transcriptome will aid in identifying the coding DNA regions, thereby regulatory sequence which are involved in the coding DNA and single copy protein coding genes, multi-gene families can be identified through functional genomics approach. And these transposable elements and tandem repeat DNAs can be used in developing a kind of markers, a kind of markers which can be later deployed in molecular determination for key traits, and thereby it is involved in genome editing and genomic assisted breeding to develop improved varieties in trees. And coming to the application of genomics in tree improvement, various applications are involved. I have quoted a few major here. Genome-wide sequences for gene mining, because in gene mining or allele mining can be done across the genome by scanning the entire genome. And also, as we earlier discussed, resequencing of the genomes will give more precise information about its genome size and it's further the genes which are involved in various process. And also genome-wide it offers assessment of genome-wide genetic diversity, and deep sequencing of transcriptomes will aid in identifying the perfect candidate genes and biological process which are involved in our trait of interest. And also it involves in fine mapping and dissection of complex traits. And these genomics technologies are also offers in genome editing technologies such as CRISPR-Cas9 technology, and also the identification of various microRNAs also can be possible through this genomics, and these are involved in effectively characterizing a genotype for our end trait and economic value. And coming to this shows genome sequencing efforts so far made in some of the important agroforestry tree species. And we know that the first tree genome was done in Populus, and the size is 434 MB. And recently, so many genomic sequences are made available in various multi-purpose tree species. Although I have so many available, I have quoted few here. And these are some of the functional genomics, some of the functional genomic efforts so far made in the traits agroforestry. So these traits including growth parameters, wood parameters, biotic stress, abiotic stress, secondary metabolites in various trees like Tectonograntis, Acerrecta, Indica, Casuarina, Salix, etc. And this slide shows the overall tree genomic efforts made on various multi-purpose tree species from 2000 onwards. That is where the Arabidopsis thaliana genome sequence was evolved from that period to till date. These are the various tree genomics informations are available in the public domain. And what are all, so far we have made so many genomics, tree genomics studies. We have studied whole genome, and what these tree genomics offered so far exactly. Okay, so these offers full genome sequencing of trees, and you can see that there are so far 2317 species were curated, and it's available in the public domain, which represent 309 genus and 16 orders. These scientific information will enhance the scientific basis on various target trait biological process which are involved at molecular level. And also these genomics in developmental genomics like wood formation, bud phenology, root development, this offers a unique idea to the breeder to build a cultivar or to architect a cultivar for our various inducers. Apart from that, these genomics offers biotic and abiotic stress responses and addressing climate scenario, changing scenario. and it gives a wide understanding at a molecular level how the response is happening at cellular and chromosomal level, and epigenomes in trees, phenotypic prediction in climate change scenario, phytogeography and phylogeny, genotype phenotype association. This is more important in tree breeding because without a help of phenotype, genotyping may not be possible. So, tightly understanding the linkage between genotype and phenotype will give a precise information about cultivar development, and it gives more idea about our target region and target genes need to be transferred. And of course, not only the tree genomics offers a productive uh, information, it also involves in conservation because we know that tree genetic resources or the germplasms are the valuable resources and need to be conserved appropriately, and it needs to be safeguarded because time to time the needs of human beings are keep on changing. So accordingly, these genetic resources can be harnessed, and it can be the needs of the humans can be fulfilled by the use of genomic technologies. And coming to the advantages of marker-assisted breeding over conventional breeding. In marker-assisted breeding, genotypic DNA markers can be obtained from any tissue of the crop plant at any stage. But whereas in conventional breeding, if you wanted to evaluate, we have to wait till a certain level, whether it can able to produce a seed or flower, whatever it may. So that is a major constraint we can eliminate. And for traits with complex inheritance, we know that complex inheritance is governed by many genes. In marker-assisted selection, each gene what it roles governs and how it can be individually can be identified and transferred separately. With the aid of marker-assisted, it is possible. And molecular markers help in selection of target alleles, as we said earlier, and selection can be made on single plant basis, where this would not be possible by phenotypic selection, and also involves in recessive genes in various plants can be maintained without progeny tests required in each generation, because heterozygous and homozygous plants can be differentiated with the assistance of codominant markers possible in the marker estimate. So these are some of the advantages over the conventional breeding. And coming to in Central Agroforestry Research Institute, we have a largest number of neem germplasm in the country, and we have a field gene bank for neem. And apart from that, we have several species germplasms at our center. And as I said, we have a neem germplasm we have collected from different states, and the exploration conducted in 1903, from 93 onwards to 2006, and we have in our field gen bank so far 170 accessions are available. These accessions are also registered and deposited in NPTGR. And we have characterized these germplasm for various traits. And recently we have characterized using molecular markers, 20 SSR markers characterized, and we found that the 170 genotypes are divided into 3 different populations. These populations are west central, southeastern, and north central according to its geographical location. And you see the population and its number of effective alleles which are ranged in this population is 2.4 to 2.88. Similarly, we have evaluated the germplasm morphologically for its oil content, kernel yield, and among the 170 we have consequently for the last 3 years we have evaluated, and we have identified some 11 prominent germplasms for high oil yield in neem. And also similarly, we have studied this genetic diversity pattern of Fungamia pinnata using SSR markers, and we have characterized this germplasm for its biodiesel properties as well, in which NRCP20, a genotype, has high biodiesel property. And the population variation, if you see that within population, 97% variability were presented. It indicates a high scope of genetic variability. And similarly, leucocephala also we have studied the genetic variability, and within population where 87% variation lies. And also we first time we have whole transcriptome we made on Pungamia for drought tolerance. So from this study we have identified various candidate genes and metabolites and biological process which are involved in Pungamia pinnata for its drought tolerance. And these are some of the field photos which shows about our field gene bank. And our field gene bank of we recently we have introduced with the collaboration with the NPPGR. We have started a project on RFID technology, radio frequency tag. We know that germplasms are valuable sources and these are perennial in nature. So these germplasms are being monitored by radio frequency based chips. These are active and passive chips. So 24 into 7, these germplasm can be monitored in terms of it's any physical damage or any kind of illegal wood logging or this kind of way. And by having a passive chip, the genotypes, whatever the morphological data we are from time to time we are monitoring, that can be entered and one can easily access and one can analyze the germplasm information from us. And with this, I would like to conclude my presentation. And once again, I thanks for the organizing team for given this opportunity to present here. Thank you very much.
Thank you so much, sir. Thank you for your presentation and detailed discussion. We'll be taking up question session now, sir. If there are any queries, kindly unmute yourself and ask. And sir, there are questions in chat box also, sir. Sir, major
Yeah,
challenges of genomics assisted breeding in forestry.
yeah. Um, yeah, as I said earlier, forest trees, trees have, it's a highly outcrossing and high heterozygosity, long joinery period. So these are the major constraints. So because of this, the tree breeding or forestry tree breeding, it takes a lot of time to develop a cultivar, and more importantly, these outcrossing behavior will cause more inbreeding when we crossing in a later in developing advanced generation. And these are all the difficulties with the help of genomics. A marker can a phenotypic and a genotypic marker can tightly link. With the help of these, can one can precisely target the target gene, and it can be in the earlier generation it can be identified whether the target gene is present or not. So we can reach our advanced generation as early as possible as compared with conventional breeding.
Anybody wants to ask? Are there other questions from the participant side? Very nice information. Please share the PPT. Can you explain some important terms apart from non-genetics? Um, Pramodji, which terms you want sir to explain? Or, is there any specific? So then how much focus given in genome sequencing is important tree?
Yeah, if you see, it's a good question. If you see that maybe 5 years before or 7, 8 years before, the tree genomics research or the study is very lesser, particularly the European countries where they studied. They have genomic whole genome information about various trees, and for last 5 years, the various institutions in India like IFGTP and so many institutions like CAFRI, so many Indian institutions, they started whole genome sequencing of various multi-purpose trees, and it's very rapidly increased number of genomes because uh, uh, in tree we know that because of the long journey period constraint and developing cultivar will take more time. The genomics only the tool which accelerates the breeding process very faster. So it's the recently many number of species. In one of my slides, you might have seen in 2022 on the time scale of genomics I have shown. So that much the fast growth has happened in pre-genomic research.
Thank you so much, sir. I think there are no further questions. So we are winding up the session. Today's lecture. Today we had 3 lectures in a row. So participants, see you all tomorrow and have a happy weekend. Thank you so much, sir. Thanks a lot.
Thank you, madam.